Now I'm going to give you a overview of very basic diffusion processing, and then we'll have a different video that will walk through the steps on Hypergator. So these instructions are using different data than we'll be using for the assignment and for the walkthrough on Hypergator, but the methods will be very similar. It's just when we do it on Hypergator, we'll need to write a little script to do it. But these instructions are from a local installation of FSL on a Mac. So the first thing we would do is convert our DICOM files that come from the scanner to NIfTY. And we can use DICOM to NIfTY. And so this command that we're using here, DCM to NIIX-Z, Y, and then dot, you would run that when you're in the directory or the parent directory of all the DICOMs. And the -Z means, do we want to compress gzip the files? And we're saying yes. And so what this does is it goes through and uses this other software called PIGS that stands for Parallel Implementation of gzip. It's software that makes the compression a lot faster than what's built into DICOM to NIfTY. And as you'll see when I go through this on Hypergator, we'll make that available to use. This is usually fast enough to do that you won't have to put this in a script, but all the other steps should be in a script, unless you happen to be running this locally on your computer if you had FSL installed. So again, on Hypergator, module load MRI CRO-GL, micro GL, but I'll walk you through that in a separate video. So we convert DICOM to NIfTY, and if we look in that directory, then we can see a number of files that were created. So in this directory, I also had the T1. We won't have the T1 for our purposes. We're just going to do the diffusion imaging, at least for this week. And so this one has the T1 files. So the NIfTY file, that's the image, and the JSON file that is created that has what we call some of the metadata that's useful. And then what's created is, for the diffusion, it would be 4 files. A NIfTY, JSON, and then these 2 text files. So these 3 are text files. Sorry for my poor handwriting on here. It's a little bit of a challenge with a mouse. But these 3 are text files. So the bval, bvec, and JSON. And we'll look at those. And certainly we'll look at them in the next video. But in this one, we'll always have screenshots of the bval and bvec. So in the bval, what this is a file that tells us what are the B values. These are diffusion weighting values. And in this particular scan, we have one B0 image, is what we call it. So there's 1 0 B value, and that indicates no diffusion weighting. There was no diffusion gradient applied. There's no weighting. So a 0 means the image is roughly a T2 weighted image. And then all the rest of these, we have 64 1000s. So that's a B of 1000. And we won't get into the physics of that, but that's a pretty common diffusion weighting that is acquired. It could be, for research, it could be as low as 800 instead of 1000. But for the most part, we're going to be looking at B1000, and then can be higher values as well, B2000, B3000, and maybe even higher, but there are some limitations. The higher your B value, the lower your signal to noise is. So typically with new scanners, especially the new one over at the Brain Institute, there's a Siemens scanner that can acquire data very rapidly. And so what will often be done is we're going to acquire multiple B values. So we'll have some zeros. We always want this background. We'll typically get a B1000, and then either 2000 as well, or a 3000, or 1000 and 3000. But that will help us when we're reconstructing the diffusion of water in the brain, even with a simple, or call simple, but a more simple model of diffusion tensor imaging that went over in the previous video, the slides. So even with that, it will it'll help have multiple B values. And so most of the newer research scans typically are acquiring multiple B values now. And then the other file, so that B value file is pretty simple. The other one is BVec, and these are your diffusion vectors. This is just these are 3 dimensions. We have X, Y, Z. So these are our X, here's our Y, and here's our Z. And those are, this is from the scanner just saying, how did this protocol measure the diffusion and apply the different gradients? And we won't get into all of this. It's not important for our class. I don't really deal with these in any of my research, but this file is, both of these files are important because this tells the software that we'll use how were the data acquired, how are the directions applied, and so then we'll be dealing with these at least as inputs to our processing. So those are the BVal and BVec files created. The JSON file is also created we won't really look at it in the next video as we're walking through the processing, but depending on the software that you're using to do processing, there are some values in here that we'd want to pay attention to. This total readout time, and either dwell time, some software you're going to deal with, but not too much. But really, what we want to pay attention to is this phase encoding direction. And this J minus, what does that mean? Well, in this case, that is what we call an anterior to posterior acquisition phase encoding. We won't get into that, but what you want to pay attention to is this value, and you can look it up. These are typical enough values that you could do a Google search for phase encoding direction J minus, and it should tell you what it is. But essentially what happens is this will tell you where the warping is. And so with a J minus here, this is an anterior to posterior phase encoding. A lot of the warping in the brain ends up right in the front. As we saw in the last video in the slides, there was a picture where the front of the brain looked concave, and it shouldn't look like that. And that was just warping that happened because of the sinuses. And so a lot of the warping with this J minus phase encoding, also called an AP, anterior to posterior, the warping ends up in the front. And if this was a J plus, then it would be a PA, a posterior to anterior phase encoding, with the warping, most of it in the back of the brain. And so a lot of why we're kind of going through this is because now what's typically acquired with new research protocols is you acquire a scan that's in your normal phase encoding direction, and that might be anterior to posterior. Sometimes it can be left to right in the brain. And then you also acquire a shorter scan with the opposite phase encoding direction. So if you have a J minus for most of your images, you can do another short scan, that would be a J plus. And so that means that you have warping in one of them, with the warping primarily in the front of the brain, other one's warping in the back of the brain, and you can use that information as you put the scans together to really cancel out the warping. So that's a lot of getting into some of the technical aspects of this that we won't deal with, but there are some values in here that can help you when you're reconstructing data. For what we're going to do, we won't have to worry about it, but readout time, phase encoding direction, dwell time is going to be a little less important, but again, it's helpful to know there are values that you can look at in here if you're doing processing, and some software will ask you, might need to specify some of these values, and if you have this JSON file, you can go in there and look and see if the value is in there. Again, it might not always be clear, what do these mean? We're not going to worry about that. So with this example, I was using some older, in quotes, diffusion data. It's good diffusion data, but it's not as good as what we can acquire now. And so the general processing of what we'll do, and this is using FSL, is the first thing we want to do is run this eddy correct. And some of these steps you can actually do in a different order, but for our purposes, we're going to do this. Eddy correct is software that's part of FSL that's used to correct for motion, and it also can correct for those eddy currents that I had talked about in the previous video, where as you're applying different gradients and then the gradients are not being applied anymore, you apply one and then you apply another gradient, what happens is you end up with these little eddy currents. Again, I had drawn, so if you have a stream and you have a little rock in the stream and the water's flowing down, and you end up with these little eddies and kind of little swirling things. In this case, water, and in the brain, it's going to be little swirling little magnetic fields that are induced, and so this can help correct for that. So we're going to apply eddy correct. There's newer tools that you typically will be using now, but just to keep it simple for our class, we're going to use eddy correct, and we'll walk through this in the next video. But after you have your nifty file, you could have that renamed, but in my case I didn't rename it because I'm going to give an output here. So this is our input, so eddy correct, and then this is our input, and then here is our output. And sorry, my handwriting looks terrible with this using a mouse. And then what this 0 here stands for is that's the reference volume that we're going to use. So we're going to, this software will align every image to the first one, and the counting starts with 0. And so with this data, with these data, this 0 image was a B0 image, so that's always a good idea, is to align to one of your B0 images. Typically you're going to acquire one of those first, and then sometimes you're going to acquire multiple B0 images, but a lot of times that'll be the first one. You don't have to align to the B0 image, it's just a good idea. So if you were looking at that B value file, the B-val file that we looked at, and it had the 0 in a different location, so maybe it had 1,000, 1,000, 1,000, 1,000, 0, and then 1,000, 1,000, 1,000. You could line up with that, and you could just say, you would count the thousands and say the 0 was the 5th scan. Then you would say, you would put a 4 there, because again the counting starts at 0. So that's how you run it. Eddy correct, input, output, and then the reference volume, and this will align everything up to that, to the B0 volume in this case, or the first volume if this was structured differently. So what do we mean by the first volume? So diffusion images are acquired. So this scan that we're using as an example here, we'll use a different one for our homework assignment, but it has 64 directions. So one B0 and then 64 diffusion gradients in different directions that are applied. And so what this is doing is going to line all of those different values up, different volumes up to the first one, and then they'll all be registered together. And so what this means is this diffusion scan is we use it for structural information about the brain. What does the white matter look like? Can we see these pathways, these major tracks, pathways in the brain? So we use it for that, but it's acquired kind of like a functional MRI. It's kind of acquired like a functional scan. And that means that you're getting one picture of the brain. So and these are acquired, there's multiple slices. So every time we're looking at the brain, we're going to look at one slice at a time, but this is going to acquire the whole brain, and then you get that picture 65 times. And so it's kind of a functional acquisition, but we treat it and it's used as a structural acquisition. So diffusion imaging, you're looking at structure, but we acquire multiple directions, and so it's again kind of like a functional scan. So eddy correct is our first tool that we're run, and then the next one that we're going to do is a software called FSL ROI, and what this is going to do is we're going to pull off from our diffusion image the B0 image. We want this as a separate image, and so what this does is we have our input. In this case, our input was our output in the previous step. So we give it input, we give our output. I'm calling it just B0 because now it's going to be a single volume. It'll be the whole brain, but it won't have the 64 diffusion directions. And then because our B0 image is the very first image in this scan, then we're going to start at 0 and take one image. Just go one step. And we'll walk through this for the homework. But what this does is it will pull off the B0 image and save it as a separate volume. And so then we can use that for some other analyses. If your B0 image wasn't the very first image in the sequence, you would know that by looking at your BVal file. Then instead of 0, if it was the fifth image, then that would be a 4, and you'd go one step. Or if you have multiple B0 images that are acquired in a scan, which can happen, then maybe you can pull them all off and average them all together, but we won't go over that. So first step, correct for motion and these eddy currents, and then we're going to create a B0 image, and then we're going to use that B0 image to skull strip. So BET that we used earlier in the semester. So you use BET, we input our B0 image, and notice how I can just leave off the .nii.gz. FSL will assume nifty or gzip nifty, and so I can just say B0, or you can type in b0.nii.gz. So input, here's the output. In this case I'm calling it DWI brain, and then telling it how much I want to pull off. Do I want to be more conservative? Do I want to be more liberal with the skull stripping? 0.3 usually works pretty well. This doesn't have to be perfect. There are other tools, other ways to make this a much better skull stripping, but for our purposes, we just need a pretty rough estimate, something that should work pretty well. And then we're going to output the mask file. So input, that's our input. Here's our output. And then this is an option that we're using to modify how it's going to skull strip. So that's our background of what we need. If we're using eddy correct, one thing we're going to need to do is do what's called a rotation of our of our BVEX. There's software that will do that automatically that'll have available on Hybrigator for you to use. And this is important because your B vectors is a description of the direction, but what you're doing with your eddy correction is you're lining all of the directions, the volumes, up with one another. And because the scan could take anywhere from 6 or 7 or 8 minutes to do, or 20 minutes. In this particular instance was a really long scan, and people aren't perfectly still. And so if you're moving your head a little bit during that time, or a lot, hopefully not a lot, but if you move your head a little bit, then you're going to have to line all of the pictures up with one another, and that's what you're doing with eddy correct. And but as you do that, that will change the BVEC values a little bit of where the diffusion gradient really is. And so that's what this rotation, we won't need to go any more in depth about it, but this is something that you need to do. Newer software tools, newer implementations of that FSL has to correct for motion will automatically do this, but this older method, this using eddy correct, which is still a valid tool, but a little bit older, and eddy correct actually works a lot better with some of these older type of data, then you need to do this rotation of these BVECs. And then the last step that we're going to walk through for our homework is what we call DTI fit. And so now this is going to be calculating diffusion tensor parameters at every voxel in the brain. And in the brain, we're defining based off of that skull stripping we did with the BET. And so again, we do our DTI fit. The instructions are down here, the command that we'll run. But we would rotate our BVECs first. So here's the script. I run that, give the input B vector, give an output. In this case, I'm calling it something like this DWI underscore EC underscore rot for rotated. And then I also input this eddy current correction log that is output automatically by eddy correct. And again, we'll walk through this in another video. So we rotate that, and then we run DTI fit. And so this is our input, is our eddy current corrected diffusion image. Give an output. This is just a prefix, so we can call this DTI. If you had a lot of people in one directory, you'd probably want to give it the subject name, but typically for this, I'll run these in separate subdirectories for each participant. And then you have your brain mask file that you're inputting, and that's the one we created with BET. And then R would be our B vectors, and B would be our B vals. B vals don't change. So you input that, and then we end up with the diffusion tensor calculations that'll give us a fractional anisotropy. It'll give us our eigenvalues, eigenvectors, all of the images like that. Again, we'll walk through this. So here's a picture. Here's the B0, what it looks like. So I said it's essentially a T2 weighted image. So CSF, other fluid, is bright. White matter is dark. Gray matter is slightly less dark. Kind of out in this area, we have the cortex. Notice the resolution, it looks very pixelated. That's diffusion tensor imaging. This diffusion weighted imaging is typically a 2 by 2 by 2 millimeter voxel. The T1 images, that are those nice structural images, are often a 1 by 1 by 1 or even higher resolution. But diffusion tends to be a little bit lower resolution. Part of that is just it's signal to noise. If you do a lower or a higher resolution, but lower value, so if we try to do a 1 by 1 by 1, that is, so a 1 by 1 by 1 voxel is 1 cubic millimeter. A 2 by 2 by 2 is 8 cubic millimeters. And so the signal to noise ratio for this one would be 1/8 as high as this normal acquisition. Normal meaning it's typical to get a 2x2x2 acquisition with diffusion data. And the problem with that is with really low signal noise, then you have to acquire more directions. So instead of 64, maybe we need to acquire 200 directions. That just takes a long time in the scanner. And so we'll typically do a trade-off and do a 2mm voxel, sometimes a little bit higher resolution if you have a little bit more time for this. But a B0 image has no diffusion weighting, and we'll look at that. This is just a single image, but this is what one of the diffusion gradients looks like. So we can see some ventricles, but if you notice, we don't see a lot of ventricle on that side, and that's just because of a head tilt. It's just where the direction of this gradient is applied. We're getting a little bit of white matter pathway that we're right here, and that's part of what we call cortical spinal tract that's coming up from the spinal cord, or connecting from the cortex down to the spinal cord, but also including pathways coming from the spinal cord up to the cortex. So we can see a little bit of structure of white matter, but this is just one of the directions. And so there, again, a video showing multiple directions. As we're going through, of course, you can see different white matter structures that can appear. We'll see corpus callosum that will appear up here. And this slice, we can see these white matter pathways a little more clearly. And so again, a lot of directions as we go through them. And then after we run that DTI fit command, then one of the images we'll calculate is an FA image. And this looks kind of noisy. By noisy, I mean we have some very bright voxels out here, really outside the brain. And that's just noise. And that's why we try to just focus within the brain. But we tend to have high FA values here in corpus callosum, where the white matter is very directional, meaning most of the white matter pathways are going one direction. We can get some other high values in some areas of the brain where, again, we have very directional values. But like I showed in the video, the slides from yesterday, or today, but posted yesterday, is there are areas right here where FA is pretty low. In some of these pixels, these voxels, we have low FA, and that isn't because the white matter isn't structured there, it's just there's a lot of crossing fibers. But this is an FA image. Another one we'll get is this colored FA image, and this is just the FA image, but then we can use FSL's viewing software to colorize it. So then we get our directions. Our red is left and right. Our blue is up and down in the brain. And then green would be sort of in and out of the image, front to back, or back to front. Again, we don't know the particular direction, we just know the directionality. And so we can color the FA image to get a little bit more information about the structure. And then we can also use the lines. And instead of a color, this has color for left to right. But we can also have a little line for each voxel. And so these lines indicate the primary eigenvector. And so those are all values that are output. The eigenvector and the eigenvalues are output by DTI fit. And so this is what we're going to do for our homework, is walk through all of this. And so this is just an overview, and then another video will be a walkthrough on Hypergator.